Before we go into that question and answer session, I'd like to give Pedro an opportunity to complete his talk. There's a storm hurricane passing by the east coast of the US, and he had a power outage when he was presenting his talk. So if he's there, he could complete the last few slides that he has to complete his talk. Pedro, are you there? I'm not sure if he, Pedro, we couldn't hear you.
I unmuted myself.
Okay, we could hear you now. We could hear you, and you could proceed with your complete your talk.
I'm sorry. Actually, I had extension cords but unfortunately, my accessory power didn't work. Okay, so where we left was there is a problem. Gastrin is important in the excretion of sodium, and thank you for listening to me again. However, there are other natriuretic hormones that are increased after sodium load. Dopamine produced by the kidney is also critically involved in the excretion of a sodium load. Aromatic amino acid decarboxylase converts L-dopa to dopamine. Renal dopamine is natriuretic. And I'll be very quick. Here is a study by Zhang et al. And here what they did was they did a renal proximal tubule selective deletion of the aromatic amino acid decarboxylase gene. And you can see in here that this is the knockout. This is the wild type kidney, but then it's not knockout intestine or in the brain, so it is proximal tubule specific. Low salt diet, no problem with blood pressure, but a normal salt diet, they knock out mice at high blood pressure, and on a high salt diet, the blood pressure increased even more. So do gastrin and dopamine interact? And here we show that gastrin and dopamine receptor type 1 colocalized in human stomach G cells. You can see here's a gastrin in here. This is the mouse stomach tissue, and here's D1 receptor, and you merge, and you can see a little bit of yellow here. So gastrin receptor increases the renal production of dopamine. Here's sodium, G cells, D1-like receptors, gastrin, and gastrin increases renal dopamine production. Food with sodium increases gastrin production and intestinal L-dopa synthesis. Carlito, you see, we borrowed from you. And gastrin increases the uptake of L-dopa. Here, gastrin enhances the uptake of L-dopa in human renal proximal tubule cells, resulting in increased renal dopamine production. Dopamine, vehicle, a little bit of L-dopa, same amount of L-dopa with a little bit of gastrin, not much happens. But when you increase gastrin in here, you can see that there's a marked increase in renal dopamine production. So increased sodium balance also increases renal dopamine synthesis independent of gastrin. And dopamine receptors and the gastrin receptor, that's CCKBR, are stimulated, and you get a natriuresis. And here to just show you that the dopamine receptor and the gastrin receptor colocalize in rat Wistar Kyoto renal proximal tubule cells. D1 receptor in green, gastrin receptor or CCKBR in red, you merge, and you can see the yellow in here. And here, low concentrations of systemically administered gastrin and D1-like receptor, phenoldipham, synergistically increase fractional sodium excretion. Again, fractional sodium excretion control, gastrin by itself, the dopamine agonist by itself, but when you put it together, you can see in here, there is a marked increase in sodium excretion. So food with sodium increases renal dopamine production that may be mediated by gastrin in here. Gastrin, L-dopa, gastrin increases renal dopamine production. Dopamine receptors are stimulated, you get a natriuresis, and blood pressure remains in normal range. But blood pressure is not increased in patients who have a gastric bypass. You can see in here, and you are kind of fat, you get a gastric bypass. This is gastric sleeve, you get rid of the part of the stomach. And in the Rowan gastric bypass, you entirely bypass the stomach. Indeed, the high blood pressure is normalized by gastric bypass in 38% in adults and 74% in adolescents. Actually, sleeve gastrectomy increases plasma gastrin and plasma glucagon-like peptide GLP-1 following a mixed meal, and GLP-1 is also not diuretic. Rowan bypass surgery prevents the increase in plasma gastrin following a mixed meal, but increases plasma glucagon-like peptide levels, and as I've already told you, GLP-1 or glucagon-like peptide is not diuretic. And as Carlito already said, the gut microbiota can participate in this process by influencing the enterochromatin cell production of serotonin, dopamine, and norepinephrine. And you can see here sodium goes into the stomach. G cells produce gastrin. Gastrin increases renal dopamine production. Dopamine receptors and gastrin receptor are stimulated. You increase sodium excretion. Blood pressure remains in the normal range, and you get help from gut microbiota, which of course I'm not going to talk to you about right now. So here I have just 3 more slides here. Thank you. When called an idiot, sometimes it's better to be quiet than open mouth and remove all doubts. So I would really like to close my mouth soon. This is Confucius lecturing his disciples. I saw this in the museum in Khufu. And part of this presentation, I have to brag a little bit, it's given by the 2015 meeting of the American Heart Association, Hypertension, when I was given this award. And this award honors excellence in research and discoveries in the field of hypertension, as well as a researcher's contribution. The selection committee assesses the candidates' impact on their field throughout their productive careers, as well as any single discovery. And 8 of the previous awardees have also received the Nobel Prize in Physiology, Medicine, or Chemistry. So the last one is, why should we not eat too much salt? Because it is a sin to eat too much salt. So there, it is a sin. Thank you for laughing. And I always end with this something by Jewish, because my mentor is Jewish. If I'm not for myself, who will be for me? If I'm only for myself, what am I? And if not now, when? Hillel the Elder and all these people help me to do all my research with funding from NIH. But the most important source of funding is NDJ, not NIH. NDJ, the initials of my wife. Thank you very much.
Thank you very much, Pedro, for such an entertaining talk. I'm sorry that you got cut off earlier, but it worked out fine. And thank you also for Carlito. And I kind of realized that what my doctor has been telling me about salt, consumption of salt, as well as sugar, are actually all true. So I think I should start following her to decrease my blood pressure levels. So
yes. Let me caution you a little bit. 16% of the people, if they eat too little salt, guess what? Their blood pressures increase. So you have to be careful.
Increases. So there's a balance. There's a balance.
There's a balance. Yes.
Yeah,
if you eat too little, it may not be very good for you.
Okay.
We can test your genes, and I'll just charge you.
Precision medicine. Yeah. Mm-hmm. With Carlito's talk, I also realized that I am missing a lot of the good food in milk, the good nutrients in milk, because I couldn't drink milk because of lactose intolerance. So I'm missing a lot of things somehow. But in any case, we are in the question and answer session, and everyone is welcome to ask a question. And I think Joel will have the first question. Joel?
Yes, thank you. Congratulations, Pedro and Carlito, for your excellent talks. My question, though, goes to Carlito. Carlito, I like your pairing of the glycome with the microbiome. I think that's really elegant. But help me out with a conundrum I have in my mind. So let's say that's accomplished. How does that then relate to what is optimal health? In other words, how do we connect or link microbiome with optimal health? Is there somebody out there who's trying to determine the proper combination or recipe of microbiome that corresponds with good health?
Yeah, this is the billion-dollar question at the moment. There's a lot of companies trying to figure out what's a good microbiome and what can we, and there's a lot of people selling probiotics. There's a lot of people, some of the things that the microbiome does is it, for example, this bacteria in infant, it acidifies the gut. And so what that means then is that when the gut is more acidic, it prevents pathogens from colonizing. And so you decrease the amount of antimicrobial resistant genes if you have a low pH. And when they've looked at babies, so there's one thing that they've been measuring forever, and that's the pH of poop. And in the early 1900s, the pH of poop was about, or baby poop was about 4.5. Today, it's closer to 7.
Wow.
And that's a big difference, but you can imagine in the adult, similar things are happening. So a lot of us are now sort of full with antibacterial resistant genes, for example. There's also other small metabolites that even affect the brain. And the problem is that a lot of the research is focused just on the microbes. They don't focus on diet. And we now know that diet has a big way of establishing what the right microbiome is. So yeah, that's a really important area of research, and I think it's hindered by the fact that we don't know the structures that are actually feeding the microbes in the gut.
Okay.
Carlito, can I add, or Joel, can I add something, because we are kind of working a little bit on that. What
Please.
We are working on is actually with the Gastrin, and we have not published this yet, Carlito, so I might need your help. For hypertension, this is not our work, is they've found that if the ratio of the firmicutes to bacteroidetes ratio is increased, that's more commonly found in hypertension. And what we are finding is that with the Gastrin knockout mice, of course, remember you need Gastrin to secrete hydrochloric acid, so you will alter the intestinal pH.
Right.
And what we've found is that knocking out the gastrin receptor will also change your microbiota. And what we've found is that you can alter the microbiota with different antibiotics. And depending upon the antibiotic, you get this and that and the other, so you can alter blood pressure. So maybe, I'm not telling you the whole thing right now, but maybe Carlito, you can help me on this project.
That's the good thing about this conference.
What's that?
That's the good thing about this symposium, the connections that we'll make.
Yes. Also, I wanted to tell you that inulin that is in the food there, actually inulin is not metabolized. And what we use inulin is that it is not metabolized, it's a small molecule, it's completely filtered by the kidney, and we use that to measure glomerular filtration rate.
Okay, so let's proceed with Gisela. Gisela has a question. Gisela?
Just a quick question, Pedro. Congratulations, first and foremost. I was wondering whether there are isoforms of gastrin and the gastrin receptor. Second question is, what is your basis for saying that hypertension is 80% kidney and 20% extrarenal? So that, and then in the case of Carlito, just wanted to know about cow's milk as a substitute for mother's milk, and is that your product? And Carlito, second is, what would you recommend for food, considering that diabetes and high glucose is a major metabolic syndrome, all over the world? So what proportion of glucose and other monosaccharides and oligosaccharides would you recommend for our diet? Thank you.
Gisela, can you repeat your question again for me, because I'm old and I was following your question to Carlito.
Yeah, so well, I remember your work on the dopamine receptors and GERT4, and you were looking at different variants of those in the populations, right? And now you're looking into gastrin. So I'm just curious whether you're also looking at isoforms or variants of a gastrin receptor and gastrin.
Yes, you are correct. We are looking also at those different variants. And we are also looking at the gastrin knockout mice, and then what we then do is to rescue the gene of interest with the gene that's carried by AAV virus. So we are working on that. That's one of the NIH grants that I have. We are not finished yet with that study. And the second question was?
The 80% renal versus 20% extra renal. What's the basis for that?
Oh, that's based on the cross-renal transplantation experiments, so that when you transplant a wild-type kidney to a hypertensive body, the blood pressure of that hypertensive body without the kidneys goes down a certain amount. And then when one now does university, they think that with their method, about 60% is renal and 40% is extra-renal.
Thanks, Pedro.
You're welcome, and thank you.
So Carlito, I think it's your turn to answer Giselle.
Yes, thanks, Giselle, for those questions. Cow's milk is what's the substitute right now for in infant formula. And if you look at cow's milk, it has some milk oligosaccharides. We did an analysis on it a while ago, and there's about a 20 to 30% overlap in the milk oligosaccharides. So and that's still pretty good. The problem is that it's very low abundance. It's about a whole factor of 10 lower. So to get the same benefit, you have to drink a lot of cow's milk. So the other thing for adults, though, is that if you think about places in the world where cows started or cows were developed, they're like in Northern Europe and in places in Africa. And if you go to those places, you find they have the highest level of people who are not lactose tolerant or who are not lactose intolerant. So they're lactose tolerant. And so it's actually lactose intolerance is actually the normal state. We're supposed to be, we're supposed to be intolerant to lactose, essentially to stop us from breastfeeding longer than we should. However, in those places where cows became available and were used, it was such an adaptation that today there are no Northern European who are lactose intolerant. So you could, for them, so if you're lactose intolerant, and you can drink, I'm sorry, if you're lactose tolerant and you can drink milk, you should, because it's a great advantage to you. Now, as for diet, what should we be eating? The problem with diabetes is that there's too much glucose. And what we should be eating then is perhaps food low in glucose. And that's why this library or this map of food will be really useful, because you can substitute things like fructose, which is not digested oftentimes if it's that different kinds of chain, or food that's high in galactose or food that's high. So yeah, so essentially you want to try to avoid glucose as much as possible. And you want to, if you do eat glucose, you want it in long, big polymeric chain that's hard for you to digest. So you can still feel full and you could eat as much as you want, but because they're not, the long polymeric chains are not digestible, it just goes right through. So I think that kind of a diet is not too far off in the future where we can sort of decide, here are the kinds of food that you should be eating so that your glucose level is not too high. So That's
Thank
it.
you.
Okay,
Thank you.
I think Asuncion Raimundo has a question for Pedro. Is Dr. Raimundo there?
Uh-oh.
Yeah, she's asking, how do you increase gastrin so there will be more sodium excreted?
Well, if you have the right gene and you put the right combination of sodium and minerals into your diet, then you'll increase gastrin secretion. And the other way to do it is to bloat your stomach, and so that if you eat something that is non-nutritious, I guess, and the stomach gets bloated, then the stress in the stomach will increase gastrin secretion.
Yeah, so you have to eat non-digestible carbohydrates.
I see.
That's what I'm saying,
Right,
yes.
right, Pedro? Yeah,
That's correct. Do you agree, Carlito?
I agree.
And that will increase gastric motility as well. And it will not only increase gastric motility, so you don't absorb a lot of your food, but will also, of your salt that you just took in, but will also tell your kidney to get rid of the salt that you've just eaten.
Very interesting. So any other have questions for the three speakers, for Dan, for Pedro, and for Carlito? I don't see anybody who is asking a question in Zoom, in the chat.
Can I ask a question for Dr.
Yeah, go
Carlito?
ahead.
Yeah. I've been eating black rice because of my high sugar level. It worked. It lowered my sugar level. But I saw that there was something different in your presentation and the constituent of black rice, Carlito. What was that?
Yeah, the structures of the glucose is different in that black rice compared to white rice. So if you don't have the bacteria to break that down, then it won't be broken down. Or it's possible that the bacteria that does break it down that's in your gut is actually the one that's helping you. So, but structurally, black rice is different in its carbohydrate than the normal white rice.
Okay, thank you.
How about brown rice, Carlito? What is that?
Brown rice, we didn't really see a difference. So I'm not, brown rice may have the kind of fiber that's not digestible because they're long, they're these, the what do you call that? The non-digestible starch. And that may be helpful in that way. And that part of it, we still haven't, it's still not that characterizable for us, determining what's digestible and what's not. But there are assays for that you can use instead of the mass spec, so that we may include that into our analysis, so that we'll have a good picture of what's digestible and what's not.
Yeah. So I suppose if no one else has any question, we're going to close the session. And there were three very informative talks from three great speakers, and I think we learned a lot from this. I'm not a chemist nor a biologist or a medical person, but I learned a lot from all three talks. And we see the future from Dan's talk. We see the future with precision nutrition from Carlito. And I learned a lot from Pedro that I should listen to my wife as well as my doctor when it comes to lowering my blood pressure. So, so shall we call it good night, good morning in the Philippines, and we'll all see each other tomorrow in some sessions.
Okay, thank you, Edson.
Thank you very much.
Thank you all.
Congratulations to all.
Thank you, Giselle. Yeah.
Thank you so much.
Thank you, Giselle.
Thank you so much.
Great talk.
Okay.
Okay. Thank you, everyone.